Okay. Once again, very good afternoon to one and all. In continuation with our microbiology class, today we will deal about introduction to microscope, or you can say basics about microscope. Basic about microscope. Before going to actual class, you should know what is the objective of today's class. Today's I will try to cover the basic of microscope and particularly important principle of microscope functioning, their parts and functions, how to handle and how to maintain the microscope in laboratory, and at the last we'll deal about the different types of microscope available in market for study. So let me start with the first, that is basic terminology. Basic terminology, in the last class I had mentioned about microorganism. I had also dealt what is meant by the micro, how smaller they are, what aid required to observe this particular organism, and we had discussed about the important microbes and their important characteristic. So now uh, today what we are going to deal, that is microscope. Microscope, you can say scope, that is a word preceding to some word that is indicating something. Last year or last class I had also I had also told you that something end with logi, that means we are studying the preceding word. So here if something end with scope, generally if you can see some terminology, it means to look at something, or it means to find out something, to view something. So that's why if you can see the terms like microscope, telescope, okay, kinetoscope, these are the scope by which we can use or we can view something. Everyone is know that this is a picture of telescope, and telescope is generally used to view the distance object like star and some other asteroids, okay. But microscope in converse to this is used to see the smaller particles. They are not visible by our naked eye. And generally in an exam, a multiple type question may ask that which instrument is used to observe the microorganism or bacteria. An option might be kept like this: microscope, telescope, kinetoscope, or all of above, like that. So you should not confuse with this one, that which instrument is used to observe the microscope. Now coming to the next, that is the history. Already sir might have covered about the history. There are two important scientists you have to remember from exam point of view as well as from your knowledge point of view. One, that is Leeuwenhoek. Leeuwenhoek in the 16th century. What he did, he is a glass maker. So he had put some combination of glasses, and he had observed that there is a small animals living in the world, and they named as animalcules. And he had observed that small creatures from the teeth, then from the rainwater, and he had given that there are small creatures that we can see by this particular instrument, and later on lot of refinement were made to improve their magnification and to overcome the deficiency that has. Later on in the mid, or you can say early 20th century, the two scientists Null and Ruska, these two scientists, they are basically physician, and they had developed one microscope that is known as electron microscope. So if you can compare the older one, that is a primitive or the first microscope, and this is considered as a latest in the sense that latest microscope of electron microscopy. So electron microscopy, then simple compound microscope, and other type of microscope, their limitation, their function, their utility, that will study in the end of this lecture. These two scientists' name you have to remember, because many times they may ask in your exam that who had discovered the electron microscope. So you should know the name of scientists who had discovered the electron microscope and who had discovered or who had seen or give the concept of microscope first time. Then coming to the principle of microscope, these two terminology, whatever I am keeping in red, they are the important. So you should know, and it must be clear in your mind. So two terminology, magnification and resolving power, it must be clear in your mind. First, that is magnification. To whom we can say magnification? Magnification is a simple word that by how much time we are capable to enlarge particular object. For example, this is a dot. If we are able to enlarge this dot for 100 times, then it looks like this, then we can say that the magnification is 100 times, or you can say 100x or 100 power. Okay, so that is a magnification, is simply how much time we are enlarging the particular image without concentrating on the quality of that particular image. So what will happen whenever we are enlarging any image, whether it is taken in our smartphone, if we are enlarged that one, that get fed up. Okay, so that fed up, that is because of less resolution power. So second concept, it must be clear, that is resolving power. So resolving power, what it means? It means that it's a shortest distance by which two particles to give distinct image. It means if you are keeping to close point, then how accurately we can see that they are the separate entity. That power of microscope is known as resolving power. So if you are increasing the magnification power, as I had earlier told that in smartphone, if you will enlarge any photograph, it gets fade up because it has less resolving power. So here you can say this is a dot, this is less resolution power, then it looks like this, that will get fed up. But if this particular instrument have high resolving power, then you can easily see the fine details of this particular dot. Or you can say at the 10x, if you are seeing the single dot, and if your good resolving power microscope, then at 100x, this single dot can be seen like this, as a two entirely different entity. So that is known as resolving power. So any microscope you will take, or while performing your practical, you should know that how much magnification and how much resolving power it has. The simple terms or simple formula for calculation of magnification is simply you have to multiply the power of eyepiece to the magnification power of objective lens. In detail we will study in coming class, or coming slides, sorry. So this is a simply multiplication of power of your eyepiece into power of your objective lens, and whatever result you are getting, that is that is the magnification. Now you can see resolving power. Resolving power has some formula D, that means resolving power equal to 0.5 into lambda divided by numerical apertures. So lambda, that is a wavelength of light we are using in our microscope, and numerical apertures, that is a diameter of objective lens. So you can say that if we need to increase the resolving power, that is a value of D, then this is a constant value, we cannot change, but we can reduce the lambda. We can reduce the lambda, means wavelength of light must be reduced, and that's why many microscopes have blue light that has a least or lower wavelength than compared to the ordinary light or sunlight. Okay. Now second thing to increase the resolving power, that is decreasing nuclear apertures. Okay, the value of Na, if we are decreasing, that also increase the resolving power, but we have limitation that beyond some point we cannot go, or we cannot reduce this numerical apertures value. So that's why we have limited resolving power and limited magnification, and based on our objective of microscope, we can select that which one is a best magnification and which one is a best resolving power. Hope these two terminology get clear to all of you, and this is an important terminology from practical as well as your theory part of your exam. Okay, now coming to the part of microscope and their function. So one by one I will go in brief about different part of microscope and how that help or how they are important for this particular microscope. So I had enlisted different part right from the top. These are the eyepiece, this is the arm, this is a stage, this is a coarse adjustment and fine adjustment. These are always in nearby each other and they have similar function. Coarse adjustment, fine adjustment. This is a base, or you can say support. Sometimes it is also referred as leg of the microscope. This is a revolving nose piece. So eye is here, nose is here, and leg is here, and this is similar to our body. Then objective lens, okay, stage clips, okay, that is present on stage, and diaphragm. So diaphragm will help to adjust the light, and at the bottom of this there is a light source that is provide the light, and it might be artificial or a natural sunlight. So these are the basic part of common student microscope, or you can say the compound microscope. Now we will see one by one in detail, and we will see the function of that this all these parts. Now the first one, that is a arm. Arm is generally the tube like structure that connect your eyepiece or this one with your base, okay, and this is generally used to hold the microscope while carrying from one place to other place. The second, that is a stage. Stage is generally, that is a place of microscope where we can hold or we can keep our slides or some object that need to be viewed. That is known as stage. And stage clips. Stage clips is generally, they are the small clips over which we can hold our slides. They are known as stage clips. Coarse adjustment and fine adjustment. Coarse adjustment, generally coarse means which are not finer, they are in bigger movement or they can move in bigger scale. So whenever we are focusing our slide in microscope, the first and foremost thing, we have to adjust the focusing with coarse adjustment knob. And once we are near to our object, then we have to move to the fine adjustment. So you have to start with a coarse adjustment. Once you are near to your view, you have to adjust with fine adjustment to refine your views. These knobs are generally present in, if you will correlate with an older radio, okay, older radio, they have two bigger knobs. We have to mute them to get the particular frequency. Then we are using smaller knob to get the clear sound. So same function is here. The bigger knob is known as coarse adjustment, that is used to get near to our view, and fine adjustment, that is to refine our view. So these are the coarse adjustment and fine adjustment. Now next, that is base. Base, I had already told that base, that is sometimes also referred as leg, and this is particular bottom of the microscope, and generally that support the entire microscope. The next, that is body tube. Okay, the length of body tube is important in magnification, and it has some limitation that we cannot go beyond that particular length. Generally 160 mm, that is a body length, body tube length, and that helps in magnification power of microscope. And in other use, that it connect your eyepiece with your objective lens. So this is act as a connecting tube between eyepiece and objective lens of microscope. The next, that is stage clip. Earlier I had already mentioned that stage clips are present on the stage, and generally that will help to hold the slide so that it will not get fall while observing the slide. The next and important one, that is a diaphragm. Diaphragm, if you can correlate with our body diaphragm, or you can correlate with our pupil of the eye, or diaphragm that are present with iris, okay, iris like. So the function of diaphragm, or whatever mechanism present in our eye, the similar mechanism happens below this stage. So what will happen, that the diaphragm is adjusted in such a way that you can minimize or adjust the intensity of light. So it depends on the type of objective lens you are using. When you require the large amount of light, you require the open diaphragm. And when you require the less amount of light, you can close the diaphragm. This work in a similar mechanism to our eye. When we are going towards the brighter side, our pupils get dilated, and that is get minimum light when there is a bright light outside. And in contrast to this one, when we are entering in the dark room, it will get open up, and it will allow to pass maximum light so that you can view the brightest image. So similar principle is happening here in diaphragm, and that need to be adjusted with the type of objective lens as well as the thickness of your sample. If your sample is larger or thicker, then you require large amount of light must be passed from that object. So you require an open diaphragm, and generally diaphragm is located below the stage, and this particular diaphragm holder that is also movable up and down motion, so you can adjust the height of diaphragm depends on your objective and your object. Now next, that is a light. Light source is also important. Generally in the older type of microscope, sunlight, simple mirror that is concave and plane mirror is located at the base of microscope, and that is simply divert the light, sunlight to your objective lens through object. In modern microscope, they have LED lights, or they have some other blue lights. They are capable to give a brighter and clear views than our sunlight. Then next, that is resolving nose piece. So this particular ring like structure below this tube and above this objective lens, that is known as resolving nose piece. The function of resolving nose piece is they can resolve in rotational fashion, and that have different hole to harbor various types of objective lens. Now coming to the second most important part, that is lens of the microscope. Lens of the microscope, they are considered as heart of any microscope. So lens, generally they are the two type of lens in any microscope. The first through which we can see, that is known as eyepiece or ocular lens. Ocular word is generally used to or referred to the eye, something related to eye. So that's why lens that is near to our eye, or we can see through the that lens, that is known as eyepiece or ocular lens. And generally they are single, double or triple. So one that is known as monocular microscope. If there is two eyepiece, then there is known as binocular microscope. If there is a three eyepiece, then that is known as trinocular microscope. Now coming the second lens, that is known as objective lens. Objective lens, they are present at the end of the tube, and they are generally close to the object. That's why it is known as objective lens. So lens that is close to our eye, that is known as eye lens or ocular lens, and lens which are close to object, that is known as objective lens. And these two lenses are important because the magnification power of these two lenses, if you will multiply, then that will give the magnification power of that particular microscope. So magnification power is depend on power of ocular lens, and once that will multiply with power of objective lens, whatever value we are getting, that is known as magnification power of that microscope. For example, here eyepiece, generally only one type of this is a or not objective lens, but this is a ocular lens. So only one type of ocular lens is generally harbor or generally present in microscope, either that is of 2x power, 4x power or 10x power. In general, 10x power is generally used in common student microscope. In contrast to this, there are 3 or 4 objective lenses are harbor in that resolving nose, and they are of different power, and their use is also different. So in next slide we will see that what is the use, but the different powers, that is 4x, 10x, 40x and 100x. So 4x, 10x, 40x and 100x, these are the power of objective lens, and this power, that is when multiplied with the power of eyepiece, then we get the magnification. So that's why power of ocular lens multiplied by power of objective lens, that give the magnification. Now objective lens, generally if you will see, they are the retractable. Retractable means they are just like turtles. If there is a hurdle, that will retract that leg and that will remain silent. So here whenever you are moving your stage beyond some limit, then this particular lens get retract, so that lens can get prevented from getting damaged. Okay, so this objective lens, that's why they are kept as a retractable lens. Now, the important slide, that is the different objective lens have different functions. So I had tried to prepare one table so that you can easily understand. So there are three important objective lens. Generally, they are known or named as low power lens, high dry objective lens power, or high or oil immersion lens. Oil immersion lens, why it is known as oil immersion? Because that required oil when you are going to view some object. So this is a power, low power generally of 4x or 10x, high power generally 40x or 45x, and oil immersion that is around 95x to 100x power. If you can see the focal length, then focal length is very like this. If it is a 10x, then focal length is about this point, 8.3 mm. If you can use the 40x, then this distance between your slide and your objective lens is only 0.72 mm. And when you are using the 100x, then the distance between your slide and object is only 0.14 mm. So this thing you should know, that whenever you are using the low power objective lens, the distance between your slide and lens is more. That's why you can view that object, or you can lower down your stage by seeing that which power you are going to use. If you are using the 100x, then it must be near to your object, then only you will get the correct view. The size is short if it is a low power. If it is has the power is increasing, the diameter is increasing, but the length is decreasing. Now ring, what I mean to say that generally this is some coded. You can say these are I have kept the photograph. If you can see that if it is a 4x that has a red ring, so this is a color coding. So color coding, that is easy to know the power of that particular lens. So from distance, if you can say that your lens or the lens you are using that has a white ring, then you can say that you are viewing in oil immersion lens. If you are using a blue color or blue ring objective lens, then we can say that you are using a 45 or 40x power. So this is generally color coded. It is given on each and every lens, and this is uniform across all the manufacturer. Now, the utility of this power is generally low power, generally used to scan your sample. Scan your sample means we have to locate our sample where it is on the slide. So whenever we are going to focus the slide, first and foremost thing, we have to scan our slide using the low power. Once we are scanning and we had find out particular locus, that this particular point we have to focus, then we can move to the higher power, and gradually we can move to highest power. So in general, the bacterial motility, fungus, and other characteristic of bacteria can be viewed by 40x, or you can say high dry power. But when you have to observe the bacteria, stained bacteria, their morphology, then you should use a oil immersion lens. So this is in brief about the comparative use and the power of different objective lens. Now, coming to the next portion, that is about the handling and maintenance of microscope. Of course, microscope is considered as one of the important instrument of microbiology laboratory, and it must be handled with care, and it must be properly maintained in laboratory so that you will get the better and clear picture for longer time. So I had narrated this handling and maintenance in one slide, and four important points you have to remember is the first, that is how to carry the microscope. If by chance if you want to displace microscope from one place to another place, then you should know that how to carry. It is very easy that which one is the correct way of carrying the microscope. Okay, this is not the correct way. You cannot take any microscope from one place to another place by holding like this. Even though this is arm, you cannot hold only by arm. You should hold it one hand with arm, and another hand should give support from the base. Okay, so this photograph is showing the correct way of carrying the microscope from one place to another place. The second, that how to view. Okay, if it is a monocular, means if there is only one eyepiece, then it's okay, you can view by one eye. But when there is a binocular microscope, there is a two eyepiece, then you must try to focus by both the eye so that you will get the single image. Many students are trying to view the object by single eye by closing second eye. That is not the right way to view the microscope or to view the object. As a single eye, if you are closing, it is not comfortable for longer time, and also it is a not correct way to get the large and bright image. The third, that is cleaning. So once you will complete your use of microscope, then always clean the lens with a clean tissue paper or muslin cloth. Whenever required, you can also use the lens cleaner solution to remove the oil from oil immersion lens. If you left as such, oil will dry up and that will spoil your lens. So it is always, even though it is not asked, then also once you will finish your work, always use the lens, objective lens of your microscope with appropriate cloth and appropriate lens cleaner. And at the last but not least, that is always cover microscope with dust preventing cover. So that cover will help to prevent the dust in entering your lens, and that will improve the lifespan of microscope. Now the last portion, moving to the last portion, that is different types of microscope. So I'm not going to deal in very depth about different types of microscope. I think my colleague may deal in different principle and different type of microscope. But just I will touch that what are the different types of microscope that earlier we will start with the bottom of slide that I had mentioned binocular, trinocular or monocular. So this is generally not the type, but you can say that how much eyepiece are present in any microscope. So if there is a two, then we can say this is a binocular. But now question will come, trinocular, what it means? We don't have any, we had never seen any person that has three eye, but it is not like that. Two is for our eye, one is for camera. That's why that is known as trinocular microscope. So now these are the basic. Beside this one, you can also learn about various terminology like compound microscope, student microscope, lab microscope. Okay, these are the general terminology used for different types of microscope based on their utility. But the important types that is based on their composition, principle, and use, they are like this. The first and simple one generally we are using in parasitology to view the large parasites, that is a simple microscope. That is followed by compound microscope. It is also known as student microscope or laboratory microscope. And generally compound microscope are monocular or binocular type. Beside this one, the next important, that is known as electron microscope. Earlier I had told you that electron microscope is an important discovery, that is by Null and Ruska, and this electron microscope had replaced the light with an electron beam, and that electron microscope had improved the resolution power to the 1 lakh time, 1 lakh गणों. Okay, generally the normal microscope has 10x eyepiece and 100x objective piece, means 1000 times magnification. But if you can go to the electron microscope, it has 1 lakh time higher magnification and resolution by replacing the normal light with electron beam, and that has different glass or lens than normal ordinary microscope. They have electromagnetic lens. Okay, so electromagnetic lens generally present in electron microscope, and that leads to improve their resolution and magnification power. Phase contrast microscope. Phase contrast microscope generally used to give higher contrast to the living organism. When we have to use unstained slide, then it requires high contrast so that we can view the different object in very correct intensity. Similarly, dark field microscopy. Dark field microscopy, or sometimes you can also heard about the bright field microscopy. Dark field means whatever background is there, that is dark, only object is bright. That is known as dark field microscopy. And generally dark field microscopy is used to see the spirochetes, okay, like listeria, leptospira generally are viewed by dark field microscopy. So There is no staining. Simply you have to take your sample. If required, you can go for concentration, and you can directly go for observing that particular sample. So that is dark field microscopy and bright field microscopy. Bright field microscopy, that is generally opposite to this one, that their object is darker and your field is bright one. Now the sixth one, that is confocal microscopy. This is an advanced type of microscopy that is known as confocal microscopy, that will take the different layer of your object, because object is 3D, that is three-dimensional. But when you are observing under normal microscope, that gives only one view of your object, so it will not give the 3D object. So to enable to get the 3D object, what scientists did, they had taken a slice of images at the different height or at the different view. They had combined all the views in the one slice, and they had make a 3D structure of that particular object. So that is a beauty of confocal microscope. The seventh and last one, that is fluorescent microscope. Fluorescent microscope, this is similar to your normal microscope. The except or difference from normal microscope is, in fluorescent microscope, instead of normal light, that has replaced with fluorescent light. So fluorescent light, what will happen? They are of the same or specific wavelength light. Once that will enter, or once that will fall on the object which is stained with fluorescent dye like rhodamine or FITC, this particular dye will emit a higher radiation wavelength or higher wavelength light. So if you are viewing any object that is stained with FITC, then blue light of wavelength you are using, then that will give green color fluorescence. So that will absorb the blue light and that will emit the green color. Similarly, rhodamine will emit the brick red color light. So fluorescent microscope, the difference is that you require to stain your slide with fluorescent dye, then only you can view that particular slide in fluorescent microscope. And second thing, that it must have specific wavelength filter that is compatible with the dye you have used to stain. So that is a fluorescent microscope. So these are the different type of microscope and their use in brief. In detail, you can study from any book. If you have any doubt, you can ask me, or in next class, sir might will touch the different aspect of microscope. Now, coming to the second last, that is a figure of electron microscope. Earlier, this microscope, this is a figure of electron microscope, TAM and SAM. Generally, these are two variant of the electron microscope. And earlier, it is so bigger that entire room is required to harbor this particular microscope. Now, with the advancement of technology, the size gets smaller and that efficiency get improved. So with this, I would like to end today's class. And before ending or before taking any question from this class, I would like to give you some assignment in continuation with our previous class, that you should enlist the scientists associated with invention of microscope. You should write the difference between resolution and magnification along with example, and different types of microscope with their use and principle. So with this, I will end today's class, and if you have any doubt, you are most welcome. You can message me, or you can put this question after completion of this one. Thanks to one and all for listening me very quietly. Thank you. Have a nice day.